Okay, welcome back to the short talks, and our first short talk speaker is Gabriele Civiletto from Nestlé Institute, and he will talk about autophagy and mitophagy modulators.
So thank you, Daniela, for the introduction. Thank you to all the organizers for this opportunity that has been given to me to present here in this exceptional conference at this stage. Today, I have the pleasure to share with you some unpublished data that we have been generated at the Nestlé Institute of Health Science, where I work as a scientist. And in particular, we are focusing on the power of the diet and natural ingredients to prevent age-related dysfunction, and in particular, I work a lot on natural ingredients that activate pathways like autophagy and mitophagy to prevent metabolic disease and age-related diseases. So this is my disclosure slide. The food, it's an important tools that we have because it's full of natural ingredients and compounds that actually has been shown to target the hallmarks of aging. The Mediterranean diet is one of the examples. It's well known to prevent a lot of health aging related diseases, but we also know that this diet is full of bioactive compounds that actually can target the different hallmarks of aging. For example, in the Mediterranean diet, there are a lot of plant-based food and olive oil, fish, etc., that have a lot of micronutrient and phytonutrient that has been shown in different preclinical studies to target a pathway like oxidative stress, autophagy, mitophagy, mitochondrial function, senescence, etc. So we have a great mediator with nutrition that we can target the hallmarks of aging. One of the biggest examples that we have in the literature is spermidine. So spermidine is one natural compound, is a polyamine that has been already mentioned in this conference. And in this study, it has been shown that a population of people in an observational study that have a high intake of spermidine, actually this correlates with lower all-cause mortality. And in particular, digging more into the food records of these people, it has been shown that most of the spermidine intake of these people was coming from plant-based products. This is just an example of the fact that plant-based products represent a source of bioactive compounds that can be used as an anti-aging for the anti-aging properties. So we focus a lot on this pathway of autophagy, which you already heard from other talk. And this is an ubiquitous pathway that is happening in the cells where the damaged material is recycled, and this prevents cellular damage, so preventing also age-related diseases. The reason why we focus a lot on this pathway is that it's well known that the pathway is at the crossroad between dietary interventions and aging. Indeed, intervention like caloric restriction, intermittent fasting, or fasting has been shown to all converge to this pathway of autophagy. And a lot of preclinical studies have shown that if we deplete autophagy, we lose the beneficial effect of intermittent fasting or calorie restriction. So in order to study autophagy, we use a zebrafish model where we basically tag the SC3 protein, which is a protein that is ubiquitous in the autophagosome, with a green fluorescent protein that is called ZS green. In this way, we are able to monitor autophagy in the body of the zebrafish larvae. Also, in order to increase the throughput of the analysis, we implemented a system where we basically put zebrafish larvae in a 96 plates, and then we treat the larvae with all compounds of interest. After this treatment, we perform eye content imaging, and after that, we also expose the larvae to ammonium chloride. The reason we do that, and this is something similar to what is done with cells with bacillomycin, is to block the late stage of autophagy in order to see the further accumulation of LC3. When basically we see an accumulation of LC3 in absence and in presence of ammonium chloride, it means that we are dealing with the compounds activating autophagy flux. And we validated this system using the best known autophagy inducer, which is rapamycin. And in this example, you can see that autophagy, that rapamycin indeed increased the amount of LC3 puncta in rapamycin-treated larvae. And when we co-treated the larvae also with ammonium chloride, we see a further increase, which indicates an increase of autophagy flux. So then we moved to a screening in a natural library, a small library, where we basically tested compound at the range between 10 and 250 micromolar of concentration, and we also co-treated with ammonium chloride. In this library, we found two molecules which are called carvacrol and thymol, which are basically isomers, and they are present in the oregano and thyme essential oil. You can see from these images, indeed, that the treatment of the larvae with the carvacrol and thymol increase the amount of LC3 puncta, and also in presence of ammonium chloride, which confirmed that thymol and carvacrol increase autophagy flux. For further studies, we only proceed with thymol since their similarity in the chemical structure. And so we want to see what happens at the metabolic level. What we did, we basically put the larvae inside in a dish, and we treat them with thymol for 4 hours. After this treatment, we measured mitochondrial respiration by Sears technology. What we observe is basically that the mitochondrial respiratory capacity of the larvae treated with thymol was decreased. However, if we wash out thymol just before reading mitochondrial respiration, we observe a rescue of mitochondrial respiration up to the level of the control, indicating that this perturbation of mitochondrial function is only mild and transient in the larvae of zebrafish. So in order to confirm whether this transient effect of mitochondria is beneficial or detrimental, we basically pre-treat the larvae with thymol at different concentration, and then we expose them to rotenone. So rotenone is a toxin of mitochondria which inhibits complex 1. Basically, what we observe is that in the larvae treated with the thymol, they were more resistant than untreated larvae to rotenone, which indicates that basically thymol increase stress response pathway to rotenone. So in order to understand if the effect on mitochondria was direct or indirect, we also titrated thymol on isolated mitochondria. We measure mitochondrial respiration in Ouroboros. So we observe indeed that increasing concentration of thymol led to decreased respiration. However, low concentration that we use as active dose, which are around 50 micromolar, only led to a mild depolarization of mitochondria. In addition, we want to see what was the fate of the mitochondria in cells that were treated with thymol. To do that, what we did, we used a mitophagy reporter where a mitochondrial protein is tagged with the GFP and RFP. Basically, what we see is that when mitochondria move to the lysosome, the color of mitochondria switch from yellow to red because the GFP fluorescence is quenched by the low pH. And indeed, we can observe that the cells treated with thymol, the amount of red mitochondria was increased, suggesting that basically treatment with Thymol, from one side activate autophagy, from another side is inducing a mild depolarization of mitochondria, which at the end lead to a recycling of damaged mitochondria, so mitophagy. So to test translatability in the mammalian system, then we went in mice. So first, we run an acute treatment where we treat mice by gavage with 2 doses of Thymol, and then we isolated proteins and mitochondria from the liver. So we observe an increase of the LC32 form in Thymol-treated mice. And in addition, we also observe an increase of the phosphoubiquitin in the serine 65 residual, which is a target of PINK1, suggesting then that both autophagy and mitophagy was increased in the mice acutely treated. In addition to test the benefit of Thymol treatment, we also wanted to see what happens to disease model, where we basically fed the mice with high-fat diet and induced liver steatosis. In addition, another group of mice was co-treated with Thymol. What we observed is that mice that were treated with Thymol show a decrease in amount of liver triglyceride. In addition, this mirrored with the decrease of the amount of lipid droplets and their size, which also was mirrored by an amelioration of the general histological features in the liver of these mice. To better understand the effect of the stress resistance, we also use C. elegans. In these models, we tested another paradigm where we basically pretreated the worms with the Thymol at 1 concentration, and then we exposed them at 37 degrees of temperature. So we test thermotolerance. We can see here that Thymol protected from the stress induced by the heat. And when we test this in a background of a PINK1 mutant worms, we see that this effect was lost, suggesting that the effect of stress resistance is dependent on mitophagy activation. The last model that we used is a mouse model of progeria. It's called SAMPATE, and this is an inbred model of mouse which show early sign of aging. We treated the mice for 3 months with Thymol 20 milligram per kilogram a day, and we measured different phenotypes. We can see here that basically the mice that were treated for 12 weeks with the Thymol show a remarkable increase of treadmill performance. This normalized to the control and to the baseline, it's equals to 2 times increase compared to control mice. We also measured force by grip strength. We see that mice that are untreated, they lose muscle force during the treatment. However, the mice that were treated with Thymol maintain a normal muscle force. This phenotyping was also mirrored by a rescue of general muscle histology and cross-sectional area. And also we measured biological age by epigenetic clock. We see a remarkable reduction of biological age in mice treated with Thymol. So to conclude, we show that zebrafish is a good model to find new anti-aging molecules that activate autophagy, and our high-content screening in vivo identified Thymol as a potent autophagy inducer. So Thymol, as a mechanism of action, improved resilience to stress associated with aging, and also rescue and protect from metabolic stress and aging. So with this, I want to thank all my colleagues at Nestlé Research and the great collaborators that we have. And thank you for your attention.
Thank you for a great talk. We have time for a quick question. Maybe then I can ask if you have a hypothesis what the target of Thymol could be, how it regulates or affects autophagy?
Yeah, so we basically believe that this thymol induces some cellular stress, and this activates stress response. So it can have a pleiotropic effect on different targets. One of these could be MPK or ATF4, so a lot of targets that are related to stress response pathways.
Okay, thank you.